It's a great pleasure to me to introduce Cristina. She's an excellent bioinformatician, currently leading the structural bioinformatics group at the Lelwar Institute Foundation. She's a great promoter of bioinformatics and responsible for the bioinformatics summer school in Argentina. She's co-founder and she was the first president of the Argentinian Association of Bioinformatics and Computational Biology. And beside her scientific career, she's an outstanding person. And personally, I was very lucky to have her as my PhD supervisor. Chris, it's an honor to have you here, and you may start when you're ready.
Thank you very much, Lynn, for your presentation. It's more than what I feel I am. So my lab is usually working in protein structure, function, and dynamics, but this work I will present you is not about that issue, but about cancer. I have another branch which give us some choice about finding patterns in mutations in cancer. So I will share the screen. So I will speak about panels and models for accurate prediction of tumor mutational burden in tumor samples. So as we all know, the cancer is a health priority in the global world. The incidence estimated is that we have about 14 million new cases a year and 8.2 million of deaths a year. So it's really a priority problem around the world. As we know, all human cells, cancer cells, carry a multitude of gene mutations that those can be potentially recognized by the immune system to work with them to kill the tumor cells. So what the amount of mutation a tumor cell has is what it is called the TMB or tumor mutation burden. From now on, you will hear me saying TMB for a shortened word, and is defined as the total number of exonic mutations in a tumor sample. And this is what the talk is about, trying to predict this tumor mutation burden. You will see why is it useful. So the tumor cell strategies to escape immune surveillance have many mechanisms, including up-regulating the expression of a protein which is called programmed cell death protein 1, PD1, and its ligand, PDL1, and also another protein which is called CTLA4. So our immune system has an immune checkpoint that in regular cases, tumor cells, but this checkpoint is meant not for tumor cell, but for antigen presenting cells. The T cell have a surface receptor called PD1 that interacts with the receptor in the other cell. And if it happens, that antigen, neoantigen, is recognized as self, and the T cell doesn't kill the other cell. But tumor cell has this strategy to make T cells not to work with it, not to kill them. But if we block these interactions with antibodies, either against PD1 or against PDL1, this interaction won't happen, so T cell will recognize these new antigens as foreign antigens and will kill the tumor cell, will control the tumor growing. That is what we would like to have in order to make the immune system try to kill the tumor. So it was seen from several clinical trials that this strategy give an overall survival of patients in many cancer types, as melanoma, lung cancer, renal cancer. So there are already several antibodies approved for being the first and second line of treatment of cancer. So they are antibodies either against PDL1 protein or PD1 protein. And those are the commercial antibodies being actually applied in cancer patients. However, not all the patients respond to this therapy. And also, in addition, the cost of the treatments are very elevated, and a lot of patients experience severe adverse effects. So in consequence, we need to have predictive biomarkers to try to divide this population of people and give the treatment to those ones who will have a benefit, a clinical benefit from this therapy. So a great number of retrospective studies have found an association between TMB, the amount of mutations a patient have, and an outcome to immunocheckpoint blockade therapy. So TMB started being a promising biomarker to predict immunotherapy response. So how do we measure the tumor mutational burden of a sample? The real value or the optimal technique will be the whole genome sequencing or whole exome sequencing. But what are the drawbacks of these techniques? Is its turnaround time, its complexity, it is costly, and have a high tissue consumption. So nowadays, is being used the next generation sequence panels, already used in clinical setting. But again, this technique has several problems. Those are that these NGS panels have not been designed to extrapolate TMB. They target genes that carry driver mutation or other clinical relevant alterations. They are not representative necessarily of the total number of mutations in the entire genome or exome, and also they are common for all the malignancy. And of course, we've seen that they are inaccurate. So with that word, we want to present a bioinformatic-based tumor-specific methodology for the selection of target genes and their panels to predict TMB. We will compare our prediction, our panel, which will be the genes we have to sequence to predict TMB, with other two very significant panels. This is the Foundation 1 panel, which is currently used, that carries 315 genes, and also will compare our panel with COSMIC genes. COSMIC is the catalog of somatic mutations in cancer, which is the biggest repository for tumor samples and their mutations. And COSMIC says that a set of genes have a set of genes that are causally implicated in cancer. Those are around 800 genes. So we will compare like this panel and imagine that COSMIC genes, this set of genes are in a panel. This is not, doesn't exist, but we will say that we can see how a panel made with these 800 genes will work, and with our selection of genes that a priori we don't know how many will we have and which genes will they be. So our workflow is the one you are seeing. We use all the samples that have been whole genome sequences or whole exome sequence from COSMIC version 84. You will see that that is an important date. So we make a lot of cleaning of the data. As you know, there are many inconsistencies, mutations that are in very few number of samples, etc. And we will try 6 strategies, taking the genes as a variable, and we will do panels with our selections, with the Foundation 1 panel at the census, COSMIC census panel of genes, and we'll do also an exon strategy with the other, and we will do it, the panel with the 3 set of genes, but taking exons as variable, and you will see later why we thought about that. We will make panels with the forward-step algorithm, and we'll make a quality measure of the panel. Some of that we put these two numbers, that if the R square should be more than 0.6, this is a statistical measure that says that above 0.6 a model is good, above 0.8 is very good. And also we'll measure the standard error of these models. At the end, we'll make an internal and an external validation of these panels. And at the very end, we will try to see if these panels can predict the immunotherapy response. So as I told you, just to remember in that very simple slide, we will make a genes strategy and an exon strategy, and we'll do panels with these three selection of genes. Our panel will start with the 22,000 genes in the human body, and also we'll have this 315 from the FO panel and 800 from the census gene panel. The census, when I say the census, is this set of protein of 800 genes that Cosmic says that are causally implicated in the disease. Why did we thought about exon? That is weird, in fact, because we always think about genes. So that was the line of thought. If we see a protein, for example, the epidermal growth factor receptor, this is the whole protein, and all the mutations, in fact, are in this domain, which is the tyrosine kinase domain. So here, the protein has around 1,000 residues. It means around 3,000 base pairs to be sequenced if we want to sequence that gene. And as we know that all the mutations are in this very small domain, but even inside that, mutations are not everywhere in the domain, but in two particular parts, which are from that domain, exons 18, 19, and maybe 21st. And if we can have a strategy that tell us that we, instead of putting the whole gene in a panel, we can put only few exons, we'll have to sequence much less base pairs. So I'll show you, this is the distribution and the number of mutations the different cancers have. And as you see, there are cancers that have a lot of mutation. Here we have something like 5,000, 10,000. And there are cancer types that have very low number of mutations, 1, 2, or 5. And also that the distribution is very different across different cancer types. Here the black numbers are the number of samples we started with working. The red numbers are the median of the TMB distribution, and the blue numbers are the numbers of genes that are shared between the different samples. So our strategy was starting from 24,000 samples from 22 tumors, we made the data cleaning. In that step, we lose 28 tumors. So we have 22 samples for 30 tumors. I will tell you later why do we lose different tumor type during the work. So we create linear regression models with a forward step algorithm, and we ask them to pass a quality control that are those numbers I told you first, that is having an R square greater than 0.6 and an error minor to the median of the real TMB. So we end up with 300,000 panels and model for 25 tumors. So with the accepted model in the previous step, as we have many, many models for each cancer type, we make what we call, what they are consensus panels and models. After having those consensus models, we made an internal validation, which is a bootstrap of 1,000 times trying these models, and we end up with almost 3,000 models that pass this internal validation for 20 tumors. But after that, we make them to pass an external validation, which is using, trying to predict the TMB with the panel of 4,000 samples that we took from literature, and also around 2,000 new samples in Cosmic 90. If you remember, we've done with the samples that were present in another version of Comic, an older version of Comic, which was 84. So, but that time we have new samples that were not introduced in the development of the algorithms. So these two set of new samples were the testing set of samples. At the end, we suggest one optimal panel and model for 14 cancer types. And we have other alternative models that are as good as the first, but we end up suggesting the optimal one. So why did we lose cancer types throughout the process? So for 21 of the 42 cancer types, we could not generate panels and models due to different causes. The first one is because some tumors have insufficient number of samples after data curation. So we think that if we have more samples, we might create also good model for predicting TMB in those tumors. Other cases, 5 cases, the models have not qualified to generate consensus model. It means it didn't pass these quality measures that we have as thresholds to say that this is a good model. And finally, consensus models for some cancer type did not pass the internal validation or the external validation. So we ended up having several models. Here you see, this is the R-square value for different models, and this is in which cancer type, and those are the number of sequences that that model has. The red line indicates, we'll have always the red line as a reference, which is around 1 megabase. That is the number of bases that the Foundation One panel has. So here, each dot is a model, and this is the R-square value for this model. And as you see, I'm seeing myself and I cannot see the results. We will speak about all the gene strategy, the census one, the panel one, and our gene selections, because the exon consistently gave worse results than the gene strategies. And as you see here, the dots are in pink, which is our exon, our gene model. And if you don't see the other colors, it's because they have no model that fulfill the quality, that can pass the quality control. So you see that for many, so I mean to be here, for many cancers, none of the other strategy, let's say the Foundation one or the census one, have a model that qualify for that is a good model. So it's why in many cancer types you don't see other colors than the pink one. For other cancer types, there are models that might qualify for as a good model to predict TMB. For example, this one is the census one, the one in here. But even though our model have a better R-square, and that's it. So what we want from a model is to have a high R-square value. That means how good is the model. But also, we want to have a low error number. And this is the same. This is the error value. Those are each cancer type. And those are the megabases you have to sequence to have those models. And as you see, for example, there are many, there are some cancers where other strategies will be good enough, but again, our models have less error. And also, if you remember that red line is the amount of bases you have to sequence to you have to sequence, sorry, is the number of bases that the Foundation One panel has, we can see that there are several models with high R-square value and low error, but you have to sequence much a smaller number of bases to have it, to have a good model. In many cases, that's true. So I'm putting together the R-square number and the error of few cancers for you to see that we want to have a high number of R-square and a low number of error. And as you see, our models tend to be better in both measures. The other important thing here is that, again, the red line is the number of bases the panel has, and these are our models, and you can see all the models, the number of bases they have. So you can see that we can make a panel with few genes, or that means few bases will be sequenced. So that means that that will be much more cheaper. Another thing we wanted to see is how common are the genes between different cancers, how much they share. And as we see, for example, if we take any type of cancer with any other, we can see that they don't share many genes in the panels. The highest value is around 40%. This is with our gene selection. With our gene selection, we can see that different cancers don't share the same set of genes in the panel, in the panels with the best models. Also, we compare our strategy, our set of genes, with the census set of genes and the Foundation One panel set of genes, and we see that our set of genes are not shared with these other two strategies. The highest value is around 20%. Is the same here. We have in our gene selection that is putting all the genes of the 14 models together, the CGC and the Foundation One panel, and you can see that we don't share many genes in our sets. So at the end, we use to publish cohort of metastatic melanoma and advanced non-cell carcinoma of lung, and because they were two cohorts of patients for which we have both the whole exome sequencing at clinical data available. And here you can see, for example, that the experimental TMB, I mean, this made with whole exome sequencing, and our predicted TMB is always very well correlated with the experimental one. Experimental and predicted have a very good correlation. This is for melanoma and this is for lung. We've seen the correlation between response to immunotherapy and our predicted TMB. And here is the experimental, how was the overall survival of the patients with experimental calculated TMB. This is those patients that have high tumor mutation burden, the ones that have low, and this is with the predicted TMB. And if you have a look at this small number here, this is how significant is the separation between this set of people. And as you see, with experimental TMB calculated, these two populations couldn't be divided. And with the predicted TMA, we were able to divide these two sets of people, the ones that will have a clinical benefit from the immunotherapy and the ones that not. And the same is true for the probability of progression-free survival, another measure of how good is a therapy. Again, the experimental TMB couldn't divide these two populations, while the predicted TMB was able to divide it. So at the end, I want to to share with you some conclusions is that we were able to predict TMB, to make panels and models to predict TMB for 14 type of tumor, including the most common malignancy, that breast carcinoma, melanoma, prostata, lung carcinoma. These panels were submitted to a rigorous internal and external validation, and our panels also range from as small as 0.23 megabase to be sequenced to 1.49, while the current use one are between 1 megabase to 3 megabase. Foundation one was 1.3, that's erroneous, 1.2, 1.3 megabases. So our results indicate that a single selection of gene is not optimal to estimate TMB to all the type of malignancy because there are too heterogeneous. So the overlap of our suggested panel and the CSC panel of genes and the Foundation One panel is always less than 20%. We demonstrate that our panels offers reliable, clinical relevant estimates of TMB, outperforming the driver-oriented next generation panels currently used. And also that our response value are in excellent agreement, the response to immunotherapy are in excellent agreement with the reported in several clinical trials. So we end up saying that we can predict TMB with high accuracy. We propose tumor-specific panels, and also that we can have very low number of genes in the panels and to be sequenced. And apart from that, they are really outperforming the currently used panels. So this work was done in collaboration with Miguel Ángel Molina Villa, which is working in the Zeus Quirón Hospital. He's the one who knows a lot about cancer. And that is a picture he likes to show, that here is the Camp Nou for the one who like football. And this is our Leloir Institute. We are somewhere here in the 4th floor. And the majority of the work was done by Elizabeth Martinez Perez, that is a PhD student, and our sponsors are the Agencia, CONICET, and we want to thank very much to that women who made that Bioinformatics and Data Science Women Congress. So that's my last slide. So to all of you, thank you very much for your attention. That's it, Annie.
Thank you. Thank you. Thanks for that nice presentation. We have time for a few questions. The first one is, did you find exons participating in alternative splicing events to be important in the models?
We didn't analyze any splicing process. We just take every exon from every gene and use them as an independent variable. If a gene has different exons coming from different splicing events, we will find all those exons and they will be inside the model. But we didn't look specifically for splicing events.
Okay. I think we don't have any more questions. We can pass to the next speaker. Thank you, Chris, again.